How's it going, ladies and gentlemen? Mr. Donahue here once again. This time we're going to take a look at the Chromatography Lab. So our objective is to use paper chromatography to separate the components of a mixture, in this case ink, and compare the retention factor of each of these components to an evidence ink sample in order to include or exclude suspect samples. Alright, so materials you're going to need would be like a 50 milliliter beaker or plastic cup, just really something to hold water, some distilled water, a dropper pipette, or like an eyedropper, a half circle template, 5 pieces of filter paper, 5 petri dishes, a pencil, and a ruler. So what you're first going to do is you're going to take your circular filter paper and your template to essentially use a pencil to draw crosshairs. Now the reason you want to use a pencil is because if we're separating pigments and ink, we don't want to use a pen and introduce new pigments and a different ink. So now once you've done that, you're going to number each quadrant with the number of pen ink that you're going to use. So this will be used for pen number 1. Then you're going to grab pen number 1 and you're going to draw right in the center about an 1/8 inch dot for your setup. So that's setup. You're going to do that for your other pens as well. Then you're going to get a petri dish and you're going to make sure the base is set down so that the filter paper, when you set it on top, which can be kind of tricky, is more or less floating over nothing. So that when you add water to it, it's not interacting with the petri dish, it's only the filter paper. So now you're going to set them up for your 4 pens. And the 5th one is going to be your evidence. So there was a ransom note, and you're going to cut up a piece of the ransom note to get the ink, and you're going to carefully place that piece of evidence in the center as if, so it looks like the rest of them. Now what you can do is you can use a eyedropper or a pipette to add a drop of distilled water, 1 drop at a time, on top of the center of these dots. So what you're aiming for is you want to keep adding water as it gets absorbed, but you don't want a puddle forming. So as that water gets absorbed, you go back, you add another drop of water, and you can start to see the ink spreads out and the pigments become separated. The reason this happens is because water's polar and the filter paper is non-polar. So as the water spreads across the paper, the polar pigments travel with the water and the non-polar pigments are going to stick to the paper. Now, some pigments are slightly polar and they're going to travel not as far as the very polar pigments. So that's how you get this separation. Some of the pigments are more polar and will travel further, where other ones are less polar, they won't travel as far, or you'll have non-polar pigments and they won't move at all. So once the water has reached about 1/4 of an inch from the edge of the paper, you're going to want to stop adding water, and now you can start to collect your data. You're going to take that circular piece of filter paper and you're going to look at which direction can you best see all of the colors. So for this pen number 1, I can see that there is a yellow, I can see that there's a purple, there is like a dark blue, and then there's a light blue, and it looks like it's best to measure them all in this direction. So I'm going to lay my ruler from the center out and start to measure how far each of those pigments went, as far as well as the water. So I can take a look. The water looks to have traveled about 3.6 centimeters, right? If I want to be precise, 3.60 centimeters. So now I want to look at all the different colors. So I got yellow, I got a purple, I got a blue, and I have a light blue. So now you just measure how far from the center did each of those go, and you're going to compare it to how far the water went. The reason that we're going to calculate the RF value is because if we added more water, the pigments are going to travel further. So if I only added 4 drops of water to the evidence filter paper, but I added 12 drops to the first filter paper, of course those pigments are going to travel a different distance, even if they were the same pigment, because I added more water. So we have to look at the retention factor, or how far did it go compared to water. So I can see yellow looks like it made it all the way out to about there, which is like 1.3 centimeters. 1.30 centimeters. The purple looks to have made it, I'm going to say that's exactly 2.00 centimeters. The blue looks like it made it I'm going to call that 6.5 centimeters. And the light blue looks like it went 3.49 centimeters. So now I'm going to grab my calculator and do a little math. The retention factor is going to be how far did the pigment go compared to how far did the water go. So centimeters over centimeters, the units cancel out. You're always going to end up with a unitless number that is also 1 or less than 1, because water's the maximum distance anything could have moved. So that's in your denominator. You're going to end up with 1 or less than 1. So I do 1.30 divided by 3.6, and I get 0.361, but I've run out of room. So I do the same thing here. I get 2 divided by 3.6, and I end up with 0.56. I do the same thing for the blue, 2.65 divided by 3.6, and I get 0.736, and I should fix these. All right, I lied to you. Wrong number of sig figs. And then the last one, 3.49 divided by 3.6, and I get 0.969. So now I can use these retention factors to compare them to the other ink samples and see if they could have the same pigments, which would mean they could be from the same pen. So here is the procedure kind of in step-by-step form. Feel free to pause it. Here are the first 4 steps, and then here are the next 4 steps. And that's really the lab. So once you get your results, you can compare the 4 suspect ink samples to the evidence ink sample and see if there's a possible match. Yeah. Hope you found it helpful. See you in class. Okay, bye.